Okay, ladies and gentlemen, we would like to welcome Professor Andrea from Nebraska University, as I remember. She was visited us last year, and this is the second time. We welcome you here at the University of Jordan. You are now one of our team. So you are in your university. Today, she will give us about her work, some of her work in the United States. You are welcome.
Thank you.
Start.
Thank you very much. Does this mic work? Okay, then I don't think I need this one. And then how do I just click? Yeah, that's great. So thank you so much for coming, and I am just so happy to be here today. I presented some stuff last year on our work, and what I'm going to do is kind of take you a little bit through that again, and then give you some information that we've done in this last year. Some of the new data that I have, it's actually some RNA gene transcription, some RNA-seq data, and hopefully I'm going to put that together with the physiology to tell you a little bit about our cow models. Some of the things that I'd like to talk about is we've sort of developed two different cow models to understand why anovulation occurs. And we're looking at two specific models, and I'll kind of go through each of those. Okay? So in addition to looking at why cow models affect cattle, we're also interested in understanding reproductive and metabolic disorders in mammals, so women as well as other species. And one of the reasons that cows are excellent models for reproductive disorders in women is they have a similar size ovary, they're monoovulatory just like women are, there's waves of follicular development that occur on the ovary, the pattern of endocrine hormones are very similar, and the length of the luteal phase and the length of pregnancy are also similar. And in cattle, we can obtain tissue, purified populations of cell types within the ovary. And I currently am in Nebraska, at the University of Nebraska. There's 3.5 cows for every person in Nebraska. So there's ample resources there for us to actually be able to utilize. I'm sorry? Most of these are beef cattle, but we do have a large dairy farm that's about 4,000 to 5,000 head as well in Nebraska. And so last year I said 4 cows for every. It's actually down to 3.5, so our population must have grown a little bit in Nebraska. So what I want to talk about today is I want to talk about our high A4 model, and when I say A4, I mean androstenedione. It's the primary androgen in female cattle, and we actually have cows that have excess androgens. About 20% of our herd appears to be that, and they do have follicular arrest. We think this may be due to inflammation. We also think it might be due to increased AMH. I'm going to talk more about inflammation and less about AMH today. We also have a model. We have developed pubertal classifications in our herd because these high A4 cows are very similar to women with PCOS, and PCOS is often detected around the time of puberty. So what we did is we went back and we said, okay, how do heifers in our herd obtain puberty? We developed pubertal from that, and we're actually looking, and we're going to look at some gene transcription in some specifically granulosa cells, which may address this and its response to FSH. Okay, so the first model that I'm going to talk about is the high A4 model. These are just some of the students that have worked on this model in the lab. including your own Muhammad Abdul-Majid. And I'm going to show some of the data that he had as well. So let's talk a little bit about this high A4 model. And specifically, I've already discussed the fact that we have about 20% that are high A4. It's similar to polycystic ovarian syndrome. They have about a 17% reduction in calving rates, so they're subfertile. They're not infertile, but subfertile. And then the other thing that I want you to think about is the fact this disorder is highly heritable. And so we're going to try to do some heritabilities on our herd related to high A4 cows. I don't have any data, but we've got some. I have a student that has emailed me while I've been here, and she's got some genotyping data that we're actually going to utilize to do a GWAS for that as well. So there's about a 21% prevalence in the general population. Daughters of women with PCOS have about a 60% chance of actually having PCOS. So this is some data that I showed you last year. This is how we identify in the follicular. Utilize that to characterize these high A4 population. So the other thing that we've also determined is normal reproductive cycle, where we have evidence that's linked with ovulation, which are these sunbursts, and we have follicular waves of growth along with normal production, which is follicles, which we believe might be partially luteinized. Okay, and we've actually shown in granulosa cells that that may be true, that these granulosa cells don't appear to be granulosa cells. They actually express genes that are luteal-related genes as well as some theca genes. So they're they've lost their identity, basically, is what we've determined. So when Muhammad got to Nebraska, he started doing some cortex cultures. He determined that these high A4 cows levels of androstenedione, okay? And I'm going to tell you that in some cases it was 10 nanograms per mil per day. Tremendous amount. If we look at it over the whole 7-day period, it's about a greater amount of androstenedione in these high A4 cows. He also determined that we had increased fibrosis. And that's also quantitated over here on the right. And he also determined that if we reduce the fibrosis, and that's also shown here, as well as we can reduce the androstenedione, and we can get follicles to progress as well. So we kind of rescued this phenotype. So just to summarize, this high A4 model, the regular reproductive cycles, they have increased androgen production in the dominant follicle and the follicular fluid. They have increased collagen deposition, which is equal to increased fibrosis. They also have increased oxidative stress. And these are all factors that contribute to follicular rest. And if we give VEGF, then we can rescue that phenotype. And so what we're trying to do with this is trying to understand what are signal transduction pathways that VEGF is actually activating, which may be rescuing this phenotype. Okay, So what I'm going to tell you a little bit, some new data that we have is we actually went and we looked at the theca from these high 4 cows, okay? So the theca cells and the conversion of cholesterol into androgens, okay? So normally we see a lot of hyperandrogenism, and it's due to cell hypertrophy and production of increased androgen. All right? So previously, we had done some work with granulosa cells from these have in that there were them is this catenin-based that can actually inhibit the Wnt signaling pathway, and it can arrest the cell cycle. So it was for genes and proteins that were being expressed in these granulosa cells that basically were not allowing the granulosa cells to function normally. And this is just some data from our previous paper that sort of shows the granulosa cell genes that inhibit cell cycle progression and that beta-catenin interacting protein, how it was higher in a high A4 population. What we wanted to do in these cells was, okay, we're going to hypothesize follicular A, that the high follicular A4 will alter the genes, also inhibit cell cycle and proliferation, but it's going to also increase steroidogenesis. So we took cows that were high A4 cows, and we compared them to our control, and we synchronized estrous cycles, and we aspirated follicles. So we collect the ovaries, and then we isolated the theca cells. And then we did some microarray, and Rachel Reith, who was sort of a postdoc in my lab for about 6 months, helped us with the RNA-seq pipeline, and she also did some IP analysis and also identified some microRNA that might be involved. So this is kind of a really crazy slide. So let me just take you through it a little bit. So here you can see our upregulated genes in these theca cells. Most of them, and this was similar in our granulosa cells, had genes that were involved in olfaction. We're not going to do anything with this. We actually just took this out because we don't have the tools to actually determine what's going on with this olfaction. Not in the bovine at present, but someday I'm going to come back to this and figure out what's going on. But if you can see, we had a lot of genes that were regulating signal transduction, and a lot were involved in steroidogenesis. Those that were unregulated were involved in mitosis and cell cycle and transcription, and there are just some examples of some downregulated genes. So it does look like these theca cells may also be arrested in the cell cycle. Now, the microRNA is always hard to look at and actually do something with because we're not treating the RNA the right way to actually get all the answers for the microRNA. This is just something that's coming along when we're doing this microarray data. And so there were a lot of microRNA that were involved in transcriptional regulation and signal transduction. So this would follow. 5 of the downregulated microRNA only had one target, that's the threshold. And some of the genes that they're going to affect are here, and they're also involved in some aspect of follicle progression. And So microRNA are going to inhibit those genes. Okay? So this was interesting. We had similar results in the granulosa. We had a lot of microRNA that actually targeted cell cycle genes and would inhibit them or arrest the cell cycle. These are just a subset of significant pathways that were affected. You can were downregulated, but mostly they were upregulated, including biosynthesis, which is going to affect that fibrosis. Signaling, which may affect steroidogenesis. So if we try to take this gene expression and talk about the physiology that's going on, we had a lot of were either enzymes that were involved in this pathway here from cholesterol to androgen, as well as some genes that may convert androstenedione to testosterone and estrone to 17-beta estradiol. Okay? So that means that the theca cells are really pumping out steroidogenesis. There's also this cell cycle arrest, and so cell cycle arrest is not allowing cell cycle progression to occur. Some of these genes that inhibit Wnt signaling and allow for activation and proliferation were upregulated, as well as some microRNA that are affecting key genes that also allow for follicular progression. So just like the granulosa cells, it looks like the theca cells are arrested in cell cycle, but they are producing tremendous amounts of androstenedione. We also saw a lot of genes that may cause collagen deposition and may be responsible for this fibrosis that we're seeing. And we also know that hyperandrogenism ovarian fibrosis, so the androstenedione itself may be upregulating these genes. And that's something that we're going to start testing now, is to see this high A4 levels in ovarian cortex cultures, does that actually cause fibrosis? And so we're also working to also cause specifically fibrosis in those theca cells. So just to kind of conclude, high theca cells can also can alter the transcriptome to regulate steroid arrest. These do explain in part the increased androgen production and fibrosis in the high A4 cows. And then we're going to try to actually isolate it and make sure that high A4, if we treat theca cells, we can recapitulate sort of this model. And that's what one of my postdoc is working on now. I'm going to stop there and ask if there are any questions on this part. And then I'm going to go into our puberty. Yes?
You test your model on other mammals other than cows?
Yeah, well, actually, that's interesting because I'm going to tell you every grant that I wrote, they said, we don't care that you have high A4 cows in Nebraska. No one else has them, right?
Did one on sheep last year. Notice any change or any difference? That this process can be reversible.
Do you think the incidence is higher in dairy or beef cattle? And has there been any research conducted on dairy cattle with regard to this high four?
cycling. These have delayed puberty. They have reduced number of calves in the first 21 days. They do have symptoms of follicular arrest that are occurring. So these are some of the individuals that are working. Sarah Nafziger mainly developed these profiles and this paper. And I'm going to tell you a little bit about my master's student right now, her work, Brooke Rudloff. So hopefully everyone understands that right? So instead of going after the earlies, what I decided we needed to do was go after the non-cycling and those start-stop discontinuous, because if we could remove them from the herd, that's about 23% of the herd. If we could remove them from the herd average was going to go up, right? And we were going to get earlier puberty that way. So that's what we decided to do. So again, Sarah Nafziger kind of did this, which has been published in 2021. And just to tell you a little bit about these non-heifers, they have decreased uterine horn diameter. They have a decreased percentage of animals. Or 5, if you don't know what that is, 5 is the most mature. They have a corpus luteum on the ovary whenever they're palpated. And tract score 4 would have a large follicle, antral follicle. 3 would have medium follicles. And maybe when you get to 2s, you're going to have an immature reproductive tract, okay? Respond to prostaglandin F2 alpha because they didn't have a corpus luteum. A decreased percentage of animals that calve within the 20. They had an increased number of antral follicles as detected by ultrasound. I'm going to tell you that women with PCOS, and I'm not saying that these non-cyclic PCOS, women with PCOS have increased antral follicles as well. And when they do studies on women, if they've been diagnosed with PCOS, they take them out of the study. So I'm really wondering what's different about these animals. It does appear that they're under follicular arrest, okay? Right. So one of the things that we did, because we had enough animals and we genotyped as well as did some whole genome sequencing, is we did some heritability estimates on our pubertal classification since we had this data. And we started genotyping in 2017. So we had it from 2017 to 2022, I believe is what we did. And so if we take all 5 levels, so all 5 pubertal classifications, we get a moderate heritability of 0.38. This is very similar to growth traits, actually, which we were very surprised that we got this high heritability. Now, if we take those that initiate cyclicity and maintain cyclicity, which are early and are typical, and we compare them to our non-cycling, which never have progesterone greater than 1 nanogram per mil, and we add them with our SSD, which are start-stop discontinuous. They have progesterone, and then they stop, and they don't have another increase in progesterone. If we look at those 2 groups, our heritability goes to 0.59, which is very high. And it got us really excited and allowed us to publish this paper, which was really good. Because when you're doing a study from 2012 and you only have about 1 paper you need to get a couple more on it. So this really excited us, and I'm going to tell you that we found a SNP, and I'm not going to talk about this data, but we found a single nucleotide polymorphism in the FSH receptor that we're actually looking at right now and trying to determine whether it may be a marker, either for the pubertal classification or the high E4. And so that's one of the things that we're doing with that now. So what Brooke wanted to do is she wanted to see how do these non-cycling they respond. They appear to be immature sexually, okay? And I'm going to show you that they also appear to have some chronic inflammation. Now, there's a lot of stuff going on here, and I apologize. I need to figure out a different way of presenting this data. But what we did is we looked at whole blood parameters about monthly from 2019 to 2020, so for 2 years. And we looked at white blood cells, lymphocytes, monocytes, reticulocytes. This is over over here, mean corpuscular volume, which is kind of the size of the red blood cells. Mine's got kind of weird on me. The summary is over here, okay? So if we look at our non-cycling and our SSD classifications, and these are sort of the delayed puberty females, we have increased monocytes and blood platelets, which is circulating inflammation, okay? There's also tendencies for different sizes of red blood cells, increased lymphocytes, greater red blood cell width to platelet count, which is also an indicator of chronic inflammation. We also have a decrease in hematocrit and a tendency for decreased hemoglobin. So they have reduced ability to carry oxygen, which may be contributing to reduced growth in these individuals as well. So in this population of delayed puberty individuals, it does look like they have some aspects of chronic inflammation. So what she wanted to ask was, how are these going to respond to FSH? And since they have this follicular arrest, will FSH actually rescue it? Okay? And sort of that's her hypothesis. She's going to evaluate granulocytic gene expression in typical and non-cycling heifers that are treated with or without FSH. This is sort of how we did it. Our stimulated, non-stimulated typical, cycling. Then we're going to have those that are stimulated with FSH. This is our protocol and our numbers for each. We overactimized these animals, we collected granulosa cells, and we did some RNA-seq just to give you some parameters. You can see that FSH stimulation a little surprising. Then we're also to look at our number and you can see that we get more greater than 7 in our non-cycling, and we also get a tendency for than 10 in our non-cycling compared to our typical when we stimulate with FSH. Okay, this is non-stimulated. So it does look FSH is causing more follicles. And remember, those animals have more antral follicles. Okay? After we took our granulosa cells, we did the pipeline for RNA-seq, and this is some of our results. You can see that we have these larger follicles that are developing in our non-cycling. And this is just to show you some that were upregulated. So in the typical heifers, so these are typical, the orange are pathways that are upregulated. Okay, the blue are those that are downregulated. So there's some signal transduction, there's some actin cytoskeleton, and there's some muscle genes. That's not really what they are, but I'll have to have Brooke put the gene names up here. But most of them are activated. Most of them are regulating, VEGF and signal transduction pathways here. Now, if we look at the non-cycling, we see very different profile, and we see many blue, much more blue here than we did previously in the typical, okay? So there's a lot of immune function that's down. If you think about ovulation, that these non-cycling females have this chronic inflammation going on. I think that FSH is trying to further and actually help with the follicular arrest. These are just the pathways stimulated versus non-stimulated that are inhibited, the non-cycling stimulated versus non-stimulated that are inhibited, so they're very different, and those that are activated. I think this is very telling. We can see a lot of gonadotropins, a lot of steroidogenesis. We see that those follicles are developing. While estrogen receptor is activated, retinoic acid receptor is also activated, and some microRNA. So this is a very different profile than what we're seeing in our typical individual. So to summarize, remember these non-cycling heifers that are stimulated with FSH have an increased number of follicles that are 7 millimeters. They have an increased number of follicles that are 10. They have a decreased number of upregulated genes related to folliculogenesis. So even though we're seeing an increase in the size of the follicle, we're not really sure that they are maybe as competent as the one from the typical. So does FSH rescue? I don't think we can really conclude that right now. But I think that FSH is trying to allow for suppression of some of this inflammation that's going on to actually allow for follicular progression. Now, is that follicular progression going to get us some oocytes that are as competent as the typical? I'm not sure. Okay? Probably not. But that's for a later date. And maybe if I'm invited next year, I can tell you about that. Okay? So right now, this is my lab. We actually came, and I don't have time to talk about the mouse data. He and Corinne actually did. And I want to thank them. I want to thank our funding sources. And I also want to thank my past lab members and everyone, my collaborators, and everyone that contributed to this work. And I would be happy to take any questions at this time.
I don't know if this is superficial a little bit, but it came to my understanding that the inflammation is the thing that's preventing follicles from development,
Preventing follicles
from
from
developing. Or is it the increased amount of oocytes and the amount of FSH is not enough to allow them all to develop?
So I think what these non-cycling and those start-stop discontinuous heifers, I think they have an increased chronic inflammation circulating, okay? And I think their bodies are trying to do something about that. And remember, reproduction is secondary. So I think that it's not allowing those ovaries to develop normally. There are many papers that have been published that talk about increased cytokine production, especially in the ovary, and how that affects gonadotropins potentially. And so I think what's happening is there is a lot, and I don't have time to tell you, there are a lot of different inflammatory factors that are being produced by the ovary. I think that that is inhibiting the ability of FSH to truly stimulate things within those follicles to allow for them to not only progress but mature.
So do you have a clue or a hint on why the inflammation happens in the first place?
That's a good question. No, I don't. But what I will tell you is their moms, so when we started out this project, we were hoping that one of these pubertal classifications were going to become our high 4 population. And our hypothesis was that because our high 4 population seems to have some of this inflammation going on, that this is what may be happening. And if we have these pubertal classifications that have inflammation, maybe they're becoming our high 4. That, I can't say that that actually happens right now. Okay? So it's very possible that it could be maturation after birth, but it also could be something during gestation as well. And we're going to tackle it a couple different ways, but I'm not going to do like a way-suckle way or a. We haven't seen much difference in the milk parameters at this point. Okay.
First of all, I want to thank you about the informative lecture that you presented. It was amazing. I have a question maybe a bit far from what you presented, but I was participating in a study that we were the relation between different energy level prepartum and the blood parameter postpartum. And we found out that high energy level prepartum will lead to high NEFA level postpartum, and that will lead to fatty liver and eventually to ketosis. And in the literature review, we found out that high blood NEFA will lead to cystic ovaries. Why do you think is that?
High blood NEFA will lead to
Cystic ovaries.
Oh. That's interesting because I would tell you in our high 4 population, we have low NEFA. We thought it was going to be high because women with PCOS. And I thought, well, maybe it's something different about the rumen. But in our high 4 population, which a lot of times they'll talk about it being cystic, but they're not they're not truly cystic, they're more persistent.
Yeah, but probably because your population was on beef, not in dairy. Because dairy, they enter negative energy balance
Right.
after calving.
Right. So saying it may be a ketosis. Metabolic factors present. What's interesting in those high A4 populations, I do think this inflammation, those high A4 cows it is a stressor. There is some oxidative stress going on. And we were hoping we could find some liver problems as well, but we haven't to this point. Any other questions or comments? We have not actually done anything related to pregnancy yet. Oh, I think it's probably happening in the hypothalamus in utero. I think there are different things that are happening in the hypothalamus. We have looked at pulsatile secretion of LH and FSH in these high A4 animals. And they have an increased LH to FSH ratio, which is very similar to women with PCOS. But we don't see the increased LH pulsatility or anything of that nature. But we do see persistent follicles, which normally occurs when you have LH pulsatility. So I don't know what to tell you there. I mean, it's a lot of work, right? Every time we do something, I'm like, oh my gosh, this is so much work, and it's this little figure. So we're trying to figure out the things that are going to get us some big answers for maybe not as much time right now. Yeah. Thanks for having me.
Okay, we would like to thank Professor أندريا Kropp. We really appreciate this scientific and knowledgeable lecture. I like the way how you connected what you have shown us last year with the new and recent knowledge and information. Also, we like the simplicity in giving our this complex information in a very simple way. For me, personally, I have learned new things from you. Thank you very much, and thank you for your coming, our staff and student. Thank you very much.